Hello everyone, very good evening to all. This is Dr. Abirami, the Physiology faculty for DBMCI MDS expert. I welcome you all to this session of most important INI-CET topic discussion. As all of you know, the recent INI-CET paper, it had many questions in physiology, the May INI-CET paper. First, let me tell you what are all the important topics that were asked in the INI-CET 2024 May paper, and also we will discuss 2023 and 2022 important topics also. So that I think will give you a good coverage of all the important topics which were asked in INI-CET and the most anticipated topics in your next exam also. So as far as I went through all the papers of the recent INI-CET, the important topics which were focused: mechanism of muscle contraction, neurotransmitters, tubular transport mechanisms, molecular motors, filtration barrier, baroreceptor reflex, memory, action potential, cardiac action potential in CVS, autonomic nervous system ANS, vasopressin, aldosterone. Two hormones, vasopressin and aldosterone. These are the important topics which were asked in INI-CET 2024 May and the anticipated topics for your next exam also. So quickly, let's go through all these topics and the high yield points from these topics. To start with, mechanism of muscle contraction. Very, very important topic. Almost every year we can see questions from muscle and nerve and this neuromuscular junction, mechanism of muscle contraction, excitation, contraction, coupling, actin, myosin, all this. So quickly let me take you through the neuromuscular junction, the major events that take place in the neuromuscular junction. This is the neuron. Presynaptically you have the neuron, the presynaptic neuron, and this is the synapse. The yellow space is the synapse, and postsynaptically we have the muscle. What are the events in the neuromuscular junction? This is an anticipated sequence based question. All these steps can be given, arranged in sequence, the events in the neuromuscular junction. So in the neuron, the first event that happens is opening of voltage gated sodium channels, and there is sodium entry. Whenever there is sodium will cause action potential. This action potential travels along the axon and it reaches the axon terminal. Action potential is a voltage. This voltage will be sensed by voltage gated calcium channels, and there is calcium entry. Once calcium enters inside, anyway you have entry of calcium, calcium will always cause movement of vesicles to release the neurotransmitter. So this calcium moves the vesicles. These are vesicles containing acetylcholine, the neurotransmitter, and this releases acetylcholine into the neuromuscular junction. What is the fate of this acetylcholine? This acetylcholine crosses the neuromuscular junction to bind to its nicotinic receptors on the muscle. There are two types of receptors for acetylcholine: nicotinic receptor on the muscle and muscarinic receptor on your organs. This is skeletal muscle. That's why it is nicotinic receptor. When acetylcholine binds to the nicotinic receptor, the same 4 steps repeat in the muscle also. What are the 4 steps that happened in the neuron? Voltage gated sodium channel opens, sodium entry. Here also in the muscle, once acetylcholine binds to the nicotinic receptor, voltage gated sodium channels in the muscle open and there is sodium entry in the muscle. What is the second step? Action potential. Muscle also, whenever sodium enters, there is action potential. What is the third step? Calcium. Voltage gated calcium channels open and there is calcium entry. But in the muscle, already a muscle has stores of calcium which are called SR. Sarcoplasmic reticulum releases calcium. And what is the fourth step? Movement of vesicles. Vesicles open up and they release acetylcholine. In the muscle, there is movement of myofilament, actin and myosin. So sodium, action potential, calcium, movement. Muscle also, sodium, action potential, calcium and movement of myofilaments. These are the steps that happen in the neuromuscular junction. If we further zoom into this muscle, when action potential goes into this muscle, let me zoom into that muscle. This is the structure of the muscle fiber. The muscle fiber has a covering layer called as sarcolemma, and this sarcolemma invaginates at certain points to form T-shaped structures called T-tubules. The sarcolemma invaginates at certain points to form T-shaped structures called T-tubules. On both the side of the T-tubule, you can see the SR, sarcoplasmic reticulum. One T-tubule along with adjacent to SR. This structure is called as a triad. One T-tubule along with two adjacent SR is called as a triad. Now the action potential goes into this triad. Once action potential moves into the triad, next let's see what are the events that are happening. So I'll show you the structure of this T-tubule now. This is the structure of the T-tubule with adjacent to SR. This is the T-tubule with the SR. When action potential moves into the T-tubule, action potential is an electrical signal. This electrical signal is sensed by DHP, dihydropyridine receptors. Where is this DHP present on the T-tubule? Sensed by DHP. The action potential, the voltage is sensed by dihydropyridine receptors on the T-tubule, and this dihydropyridine receptor moves inside. If this is dihydropyridine, it will move inside and it will touch ryanodine. Once DHP touches ryanodine, ryanodine will open up. What is ryanodine? It is the calcium channel present on sarcoplasmic reticulum. So when action potential arrived, this electrical signal is sensed by dihydropyridine receptor. It's a voltage sensor, and this dihydropyridine receptor moves inside. Electrically it is sensing the action potential. Then it moves and touches ryanodine. This is mechanical. This was a question. Electrically followed by mechanical connection. Yes, the dihydropyridine senses the action potential, which is an electrical signal. First event is electrical signal in the T-tubule, and this DHP moves and touches ryanodine. Touching is mechanical. So the T-tubule action potential is linked by electrical coupling followed by mechanical coupling. DHP moves inside, touches ryanodine. Ryanodine opens up to release calcium, and this calcium acts on the myofilaments, actin and myosin. And actin and myosin, when they move, the muscle contracts. So this is the mechanism of muscle contraction. Further, if we zoom into the structure of this actin and myosin, we can see this structure there. There are two Z lines. So the lines that you see, these are Z lines. The space between two Z lines is called as a sarcomere. So this entire length is a sarcomere. The sarcomere has thick filaments and thin filaments. The thick filament is called myosin. So the red ones that you see, they are the myosin. The thin filaments, the blue lines, are the actin. Both actin and myosin are attached to the Z line. Actin is attached to the Z line by alpha actinin. Myosin is attached to the Z line by titin. When you look at this structure into the electron microscope, it appears as dark and light bands. What is this dark band? The entire myosin is dark because it is thick filament, that is called A band. So this is the A band. In the center of the A band, there is a small area which is comparatively lighter. So in the center of the dark band, there is a lighter band. Why is it lighter? Because there is no overlap of actin. Look at this, actin ends here itself. So there is a center space where there is no actin. So it will appear comparatively lighter. So this is the lighter zone, H zone. The lightest zone is the I band, because I band contains only the thin filament actin. So this is the lightest zone, I band. This is the electron microscopic structure. When SR releases calcium, when sarcoplasmic reticulum releases calcium, this calcium binds to troponin C, which is present on the actin. I will show you the structure of actin. The purple colored balls that you see, they are all actin filaments. On this actin, you can see a yellow colored rope, which is called tropomyosin. It's a rope. On tropomyosin, the green colored balls are troponin C. So the purple dots are all actin. On actin, the rope is tropomyosin. On tropomyosin, the green colored balls are troponin C. When SR releases calcium, this calcium binds to troponin C. Once calcium binds to troponin C, the rope is lifted off. Tropomyosin is lifted off. When the rope is lifted off, this will expose the myosin binding sites on actin. Initially, the rope was covering this myosin binding site, but once the rope is lifted off, this myosin binding site is exposed. Once the myosin binding site is exposed, the myosin head pops up and it tries to bind to the myosin head. Myosin head binds to the myosin binding sites with the help of high energy ATP. And this will pull all the actin together. Once the myosin head binds to the myosin binding site on actin, so it's like this. Myosin head binds to actin, it will pull actin. Once all the actin come closer like this, the whole muscle will contract. This is called power stroke or sliding filament theory. How ATP will pull all the actin filaments closer like this. Power stroke or sliding filament theory. When all the actin filaments move closer, the whole muscle contracts. This is the entire mechanism of muscle contraction. Next, let's move on to neurotransmitters. Another important topic from which questions were asked recently, neurotransmitters. There are two types of neurotransmitters, excitatory neurotransmitters and inhibitory neurotransmitters. The major excitatory neurotransmitter is glutamate. The major inhibitory neurotransmitters are GABA and glycine. To be very specific, the major inhibitory neurotransmitter in the brain is GABA and the major inhibitory neurotransmitter in your spinal cord is glycine. There was a question about excitatory and inhibitory neurotransmitters. Over to tubular transport mechanisms in kidney, renal physiology, the transport in the tubular membrane, very important. In the mechanism of urine formation, when substances are passing into the glomerulus inside the nephron, from the glomerulus they move into the tubule. In the tubule, proximal tubule, loop of Henle, distal tubule, collecting duct. There are 4 parts in the tubule of the nephron. What are all the transport mechanisms happening in these 4 parts of the tubule? That's what we are going to see. Let's start with the proximal convoluted tubule. The first part of the nephron is proximal convoluted tubule, PCT. In the proximal convoluted tubule, 60% maximum reabsorption of solute takes place and maximum reabsorption of water also takes place. So this is very important point. Maximum reabsorption of solutes and water take place in which part? PCT. 90% bicarbonate reabsorption takes place here. So maximum bicarbonate reabsorption also is over in the PCT. Active vitamin D formation happens in the PCT. From the proximal convoluted tubule, now the filtrate goes into the loop of Henle. Loop of Henle has a descending limb and an ascending limb. What is the function of descending limb of loop of Henle? Descending limb of loop of Henle has only water channels, aquaporins. So only function of descending limb is water reabsorption. This segment is impermeable to salts. Only water can be reabsorbed. From the descending limb, now the fluid goes into the ascending limb of loop of Henle. What are the functions of the ascending limb? Ascending limb has thin portion and thick portion. The thick ascending limb is very important. The thick ascending limb. There's one important channel here, NKCC, sodium, potassium, chloride channel. It will transport sodium, potassium and 2-chloride. Blocker of this NKCC is loop diuretic. What is a loop diuretic? Furosemide. Furosemide is an example for loop diuretic. And one disease related to this segment is Bartter syndrome. Mutations of different channels in the thick ascending limb can result in Bartter syndrome. From the thick ascending limb, now the fluid enters into the early distal convoluted tubule. Distal convoluted tubule has early portion and late portion. Early DCT has special features. Early distal convoluted tubule. Sodium chloride reabsorption with the help of aldosterone. So here only we have the effect of first hormone aldosterone acting on the early DCT. And one disease related to this portion is Gitelman syndrome. In the thick ascending limb, Bartter syndrome. In the early DCT, Gitelman syndrome. Cortical collecting tubule and medullary collecting tubule. Collecting tubule is the last part of the tubule. Cortical collecting tubule. What are the functions? The hormone aldosterone helps in three important functions happening in the cortical collecting tubule. What are the three important functions helped by aldosterone? Acidification of urine, sodium reabsorption, potassium secretion. All three functions are helped by aldosterone. And there is also another hormone, ADH, antidiuretic hormone or vasopressin, which helps in water reabsorption in the cortical collecting tubule. So first three functions helped by aldosterone, and the last function, water reabsorption, is helped by ADH. So we have two hormones acting on the cortical collecting tubule, aldosterone and ADH. And there's one disorder related to this collecting tubule, cortical collecting tubule, and this is called Liddle syndrome. Liddle syndrome. So the thick ascending limb, Bartter syndrome. Early DCT, Gentleman syndrome. Collecting tubule, it's a Liddle syndrome. Medullary collecting tubule. There's only one function, water reabsorption with the help of ADH. So that is about the important points related to tubular transport mechanisms. Over to molecular motors. There was a question on molecular motors. What is a molecular motor? Motor means it can move something else. That is a molecular motor. In axonal transport mechanism, we had learnt about kinesin, dynein. In muscle, we learnt about myosin. Kinesin and dynein, they are involved in axonal transport mechanisms, and myosin is involved in muscle contraction. All these three are called molecular motors. Why they are called molecular motors? Because they can move something else. Kinesin and dynein will help to move substances on the axon. Myosin will help to move actin. That is why kinesin, dynein and myosin are called molecular motors. Do not confuse with actin here, because actin is not a molecular motor, because actin cannot move anything else. Actin will move on its own. Actin cannot move anything else. A motor means it should move something else. Actin is not a molecular motor. Myosin is a molecular motor because it moves actin. Actin is not a molecular motor. It will move on its own. Over to filtration barrier. Again, in renal physiology, there was a question on filtration barrier. What is this filtration barrier? In the structure of the nephron, this is the glomerulus and this is the tubule. The glomerulus is like a filter which will filter your plasma. So in glomerular filtration rate, GFR, who was doing this filtration? The glomerulus, the capillaries only are doing this filtration. So at the surface of this glomerular, the glomerular capillaries, there is a filtration barrier. Look at the filtration barrier. This is the nephron. If you zoom into the structure of this glomerulus, you can see this structure. You can see the capillaries, glomerular capillaries with the this is a glomerular capillary with the endothelium, and you can also see the glomerular filtration barrier and foot shaped processes called podocytes. If I further zoom into this structure, so it will look like this. So this has, if this is the glomerulus, the glomerular capillary, the capillary has an inner wall, endothelium. Inner wall is called endothelium. Outer wall is called basement membrane, glomerular basement membrane or the basal lamina. And outer to that you have the podocytes. So inner wall is endothelium, outer wall is basal lamina or the basement membrane. Outer to that you have the podocyte. So these three layers form the filtration barrier wall. Because they are a wall, through them filtration cannot take place. In between them there are filtration slits through which filtration will take place. So inner wall, outer wall and the podocytes, endothelium, basement membrane and the podocytes, they form barriers for filtration. They are like a wall. So the barriers for filtration are formed by endothelium, basement membrane and the foot-shaped processes, podocytes. Baroreceptor reflex. What is baroreceptor reflex? Baro means pressure. Whenever there is any change in pressure, it will be sensed by your baroreceptors. For example, if there is an increase in blood pressure, it will be sensed by the baroreceptors. Where are they located? On the arch of aorta, which is called aortic arch receptors, and on the carotids you have carotid sinus receptor. So the aortic arch receptor and the carotid sinus receptor, they are baroreceptors. They contain the baroreceptors. They can sense the pressures. Any change in BP, if blood pressure increases, it can be sensed by these receptors, and they send two nerves, glossopharyngeal nerve and vagus nerve. From the carotid sinus, it is the glossopharyngeal nerve. Glossopharyngeal nerve is the 9th cranial nerve, and from aortic arch receptors, 10th cranial nerve, vagus nerve. So the 9th cranial nerve and the 10th cranial nerve, they take this impulse into the medulla. So this impulse reaches the center for regulating BP. Where is the center for regulating BP? On your medulla, brainstem. From the medulla, you have nucleus tractus solitarius. From nucleus tractus solitarius is the center for regulation of BP. In nucleus tractus solitarius, there are two functions. So this can inhibit the sympathetic nucleus, which is your VMC, vasomotor center, and it can activate the parasympathetic, which is your vagal nucleus. So sympathetic is inhibited and parasympathetic is activated. What will happen if you inhibit sympathetic? BP will fall. Therefore, there is a fall in the blood pressure. That is how any increase in BP through baroreceptor causes a fall in BP. This is called baroreceptor reflex. Very important reflex for regulation of BP. It is a short-term regulatory mechanism for regulating your BP. What are the other short-term regulatory mechanisms? Baroreceptor reflex, chemoreceptor reflex, and CNS ischemic response. These are the short-term regulatory mechanism. Which is the intermediate regulatory mechanism? Capillary fluid shift. Which is the long-term regulatory mechanism of blood pressure? RAS, renin angiotensin aldosterone system. RAS is for long-term regulation of BP. Memory. What is which is the most important organ involved in memory? Hippocampus, because hippocampus can convert short-term memories into long-term memories. Hippocampus plays a very important role. What are the types of memory? Explicit memory and implicit memory. What is explicit memory? Explicit memory is otherwise called declarative memory. Implicit memory is otherwise called non-declarative memory. Do you remember this in the regular classes? I told you explicit memory is like a Viva exam. In a Viva exam, if the examiner is asking you a question, you have to declare your answers. You have to say the answer. You have to express the answer. So it's like a Viva exam. Declarative memory, explicit memory. Express the answer, declare the answer. Implicit memory is like a practical exam. In a practical exam, no need to say, but you have to do it and show. So non-declarative, no need to say the answers, but you have to do it and show. Practically for motor skills, it is implicit memory. Declarative memory, this is of two types. Semantic memory, if you are able to remember facts, it is semantic memory. And episodic memory, if you are able to remember events, that is episodic memory. Implicit memory, this is of two types. Implicit memory is especially for motor skills, practical works, habits, techniques. All this is by implicit memory. The different types of implicit memory or the mechanisms of implicit memory. Priming. Priming is learning with the help of clues is priming. Mnemonics, storics, all this is priming. Associative conditioning. Associative conditioning examples are Pavlov's experiment, the reward centers, punishment centers, activation of reward center, activation of punishment center. All this falls under associative conditioning. In associative learning, there are actually two types of associative learning. Operant conditioning and classical conditioning. Operant conditioning means activation of reward centers and punishment centers. Classical conditioning is your famous Pavlov's dog and the bell experiment. Every time you ring the bell, give the food to the dog. The dog is learning that if you ring the bell, you'll give him the food. That is classical conditioning. So that is all associative learning. Then we have procedural memory. Procedural memory is especially for procedures. When you want to learn a procedure like dancing or swimming, any procedure is learnt by procedural memory. And the other one is non-associative learning. What is non-associative learning? Not associated with any of this. Just you learn by basic mechanisms of learning. Not associated with clue or any conditioning, any reward centers, nothing. Just you learn by basic mechanisms, that is called non-associative learning. These are the different types of memory. And remember, hippocampus plays a very important role in memory. Over to nerve action potential, a very important topic. Every time you can see questions on nerve action potential. Quickly let me take you through the nerve action potential. In nerve action potential, you have membrane potential in millivolts on y-axis, time in milliseconds on x-axis. The relation between membrane potential changes along with time is called as action potential curve. You take a cell and you stimulate the cell using an electrode. You give a current, you give a stimulus to the cell. It will show all this change. That is called action potential. The first stage is RMP, resting membrane potential. What is resting membrane potential? Due to potassium ions, because at rest the cell is most permeable to potassium ions only. That is why RMP, resting membrane potential, depends on potassium ion. Then we have the small yellow line. This is called local potential. Local potential is because of one or two sodium ions, few sodium ions, not many sodium ions, just one or two sodium ion opening will cause local potential. Once local potential reaches the threshold or firing point, from there it will fire an action potential. So from there, this is called the actual action potential. The first stage where positivity increases, that is called depolarization. What is depolarization? Due to sodium influx, because of voltage gated sodium channels opening. So it will go to positivity, it will even cross 0, go to plus 35 also. And after that it should come back to negativity. That is called repolarization. Returning back is repolarization, which is due to potassium efflux, potassium moving out. This is because of opening of voltage gated potassium channels. It does not stop at RMP, it is going beyond RMP, which is called hyperpolarization. Hyperpolarization is due to slow leaky potassium channels. Repolarization is due to voltage gated potassium channel. That is different. They are gated by voltage. Hyperpolarization is due to slow leaky potassium channels. These are leaky channels. These are not voltage gated, they are leaky channels. That was nerve action potential. Now cardiac action potential. What is the difference between nerve action potential and cardiac action potential? Cardiac muscle has two type of layers in it. It has cardiac ventricle cells, the normal myocardial cells, and it has the SA node cells in it. So there are two types of action potential for the heart: cardiac ventricle action potential and SA node action potential, which is the pacemaker potential. I'll tell you the special features of these. Cardiac ventricle action potential. In cardiac ventricle action potential, depolarization is due to sodium influx, same as nerve. Repolarization is due to potassium efflux, that is also same as the nerve. But in between depolarization and repolarization, there is a plateau phase, a flat phase. And this plateau phase is because of opening of L type of calcium channels, long lasting calcium channels. When these calcium channels open, there is calcium entry, and that is responsible for the plateau phase. That is why calcium channel blockers will act on this phase only. That's how calcium channel blockers like nifedipine, verapamil, diltiazem, they act as antiarrhythmic drugs by blocking this phase. And the RMP of cardiac ventricle is minus 90. Over to SA node action potential, which is called pacemaker potential. In SA node action potential, first difference is depolarization is due to L type of calcium channel. First of all, depolarization in any other part of the body is due to sodium influx. But here alone it is due to calcium influx. That is a special point here. and this slopey phase is called pacemaker potential or slow diastolic depolarization phase. What is the reason for the pacemaker potential or slow diastolic depolarization phase? Because of three channels: IF, a funny current. IF means a funny current. TCAL, transient type of calcium channels, and NCX, sodium calcium exchanger. IF, TCAL and NCX are responsible for this slopey phase. So phase 4 is called pacemaker potential, that is due to IF, TCAL and NCX. Over to important points in autonomic nervous system. In the organization of autonomic nervous system, you see that there is a ganglion which is called the autonomic ganglion. The nerves that are present before the ganglion are called as preganglionic neurons. This is preganglionic autonomic fibers. And those that are present after the ganglion are called postganglionic autonomic fibers. Preganglionic and postganglionic. The preganglionic fibers, at their nerve terminal, they release neurotransmitter acetylcholine. So the preganglionic autonomic fibers always release acetylcholine only. Whether it is sympathetic or parasympathetic, it will release only acetylcholine. But the postganglionic neurons at their nerve terminals, they release two types of neurotransmitters. If it is a sympathetic postganglionic, then it will release norepinephrine as the neurotransmitter. If it is a parasympathetic postganglionic, then it would release acetylcholine as the neurotransmitter. And there is one exception to this, where a sympathetic postganglionic, instead of norepinephrine, produces acetylcholine. Where is this? Yes, sweat gland. This is the only exception where a postganglionic sympathetic, instead of norepinephrine, releases acetylcholine in the sweat glands. Over to antidiuretic hormone or vasopressin. Which gland produces antidiuretic hormone? Actually produced by the hypothalamus and stored inside the posterior pituitary. Produced by hypothalamus and stored inside the posterior pituitary. It's a hormone of the posterior pituitary. What are the functions of ADH? It helps in water reabsorption. The most important function is water reabsorption. That's why it is called antidiuretic. Diuresis means water going out in urine. Antidiuresis means water not going out in urine. Water is reabsorbed into the blood. So antidiuretic hormone, what is the function? Water reabsorption. The other name is vasopressin. Vaso means blood vessel. Pressin means constriction. So it will cause blood vessel constriction. So what is the other function? Vasoconstriction. So antidiuretic hormone or vasopressin helps in water reabsorption and vasoconstriction. Aldosterone. What is the function of aldosterone? Already we saw. Aldosterone acts on the cortical collecting tubule of the nephron to help in acidification of urine. How is it helping in acidification of urine? By H plus secretion into the urine. It will secrete acid into the urine and it can cause sodium reabsorption and potassium secretion. So three important functions of aldosterone: H plus secretion, sodium reabsorption and potassium secretion. Don't confuse ADH with aldosterone. Aldosterone is called as salt hormone because it helps in salt reabsorption. That's why it's a salt hormone. But ADH, what is ADH? Antidiuretic hormone. What is the function of this ADH? Antidiuretic means it helps in water reabsorption. Other name is vasopressin. Vasopressin means it helps in vasoconstriction. Next, let's see the important high yield topics from INICET 2023. So just now we discussed all the important topics from the latest INICET 2024 May paper. Now let's see 2023 the high yield topics are very few questions actually. auditory pathway and anticoagulants. Those were the high yield topics in 2023 NICT. Auditory pathway. What is auditory pathway? In the mechanism of hearing. It's called as the auditory pathway. What are the steps in this auditory pathway? The mnemonic is ecolema. It was a repeat question. I think 2 or 3 times the question was asked. What is this ecolema? What are the steps in the auditory pathway? E for 8th cranial nerve. C for cochlear nucleus. So from the 8th cranial nerve, the impulse goes to the cochlear nucleus. From the cochlear nucleus to superior olivary nucleus. From superior olivary nucleus to lateral lemniscus. L for lateral lemniscus. E for 8th cranial nerve. C for cochlear nucleus. O for olivary nucleus. L for lateral lemniscus. And I for inferior colliculus. M for medial geniculate body, and A for auditory cortex. That is what is the meaning of this ecolema. The mnemonic, ecolema is the mnemonic. From 8th cranial nerve, how does the impulse reach the auditory cortex? Ecolema. Anticoagulants. 2 types of anticoagulants. Oral anticoagulants, warfarin and dicomarol. What is the mechanism of action? They block vitamin K. If you block vitamin K, you block vitamin K dependent clotting factors. What are the vitamin K dependent clotting factors? 2, 7, 9 and 10. So the mechanism of action of oral anticoagulants is by blocking 2, 7, 9, 10. The other injectable anticoagulant is heparin. What is the mechanism of action of heparin? Heparin blocks the conversion of prothrombin into thrombin. This is an important step in clot formation. So heparin will block the conversion of prothrombin into thrombin. That's why it becomes an injectable anticoagulant. Over to NICT 2022 paper. The high yield topics were conducting and respiratory zones in the respiratory, general visual afferents and special visual afferents, cones and visual acuity, myopia and hypermetropia, functions of lungs, pain fibers, oxygen hemoglobin dissociation curve. Let's discuss quickly all this. Conducting and respiratory zones. Weibel's classification. The entire respiratory zones are divided into, or the respiratory generations are divided as 0 to 23, starting from trachea to alveoli. The first 16 generations, 0 to 16, is called conducting zone. It contains 150 ml of air, which is called anatomical dead space. From 17 to 23, they are called respiratory zone. Why 0 to 16 is called conducting zone? Because there is no alveoli, respiration cannot take place, just conduction of air only can take place. That's why it's called conducting zone. 17 to 23, why is it called respiratory zone? Because it contains alveoli, the alveolar sacs, and alveoli helps in respiration, gas exchange. That is why 17 to 23, where alveoli is present, is called respiratory zone. What are the important points about this conducting zone? Conducting zone contains cilia. What is the importance of cilia? Ciliary motility disorders. If the cilia is not moving, it can result in cystic fibrosis, Cartagener's syndrome. 2 important ciliary motility disorders. The respiratory zone. Respiratory zone has 2 types of cells. Type 1 pneumocyte and type 2 pneumocyte. So they are actually present on the alveoli, type 1 pneumocyte and type 2 pneumocyte, alveoli of the respiratory zone. What is the function of type 1 pneumocyte? They help in gas exchange. Type 2 pneumocyte, they produce surfactant. Respiratory zone also contains dust cells or alveolar macrophages. Macrophages do phagocytosis. So dust cells are phagocytic cells. They are also present in the respiratory zone. Over to general visceral afferents and special visceral afferents. What does this mean? General visceral afferents, they carry all the impulses, informations from visceral means organs, internal organs, glands and blood vessels. The afferent means carrying signal from organs, glands and blood vessels into your brain. Special visceral afferent means it's very special, only for the taste. Only for the taste sensation you have special visceral afferents, which is carried by facial nerve, glossopharyngeal nerve and vagus nerve. So the special visceral afferents carried by facial nerve, glossopharyngeal nerve and vagus nerve. General visceral afferents are signals carried from organs, glands and blood vessels. Only for taste it is special. Visual acuity and cones. There are two types of photoreceptors, rods and cones. Rods have an extreme sensitivity to light. Cones have less sensitivity to light, but cones have a high visual acuity. And cones are also responsible for color vision. So cones, 2 points, high visual acuity and high color perception is by cones. And this cone is highly concentrated in the fovea. Fovea is rich in cones. So fovea has high visual acuity and high color perception. Over to the visual refractory errors. This is the normal eye, how images form on the retina, how it's falling exactly on the retina. If the image falls in front of the retina, like this, it falls in front of the retina, then it is called myopia or short-sightedness. Image is falling in front of the retina, short-sightedness. If the image falls beyond the retina, this is called hypermetropia. Image is formed behind the retina, that is hypermetropia. So these are the two different types of refractory errors. A word about the functions of lung, synthetic functions of lung. The important substances synthesized in the lung are histamine, prostaglandins and kallikreins. The important substances which are removed by the lung are prostaglandins, serotonin, norepinephrine, acetylcholine and bradykinin. So can you see, prostaglandins are both synthesized and removed by the lung? Yes, I think that was the question. Which is the substance which is synthesized and also removed by the lung? Prostaglandins. Over to two types of pain fibers. There are two types of pain. Fast pain and slow pain. Fast pain is carried by the fast A delta fibers. Slow pain is carried by the slow C fibers. The other name for fast pain is first pain or epicritic pain. The other name of slow pain is second pain or protopathic pain. What are the examples for fast pain? A needle prick injury or an electric shock injury. What are the examples for slow pain? Slow pain means dull aching type of pain. Yes, abdominal pain. Any abdominal pain or pain coming from inside from your viscera inside. It is called visceral pain. Abdominal pains and visceral pains are examples for slow dull type of pain. Oxygen hemoglobin dissociation curve, the ODC curve. I think you all might have learnt about the normal ODC curve. The red line that you see, this is the normal ODC curve. It can be shifted to the right like this or to the left. What are the causes for right shift of the curve? R for right shift of the curve. R for release of oxygen to tissue. Whenever there is a need to release oxygen to tissue, the curve will shift to the right side. When the tissue will need oxygen? In hypoxia, anemia and in exercise. What are the factors which cause right shift? Right means good. So we'll put all positive factors. Increase in H+, increase in CO2, increase in temperature, increase in 2,3 BPG. All this causes right shift. What are the factors which cause increase in 2,3 BPG? Growth hormone, thyroid hormones and androgens. And this right shift is called Bohr effect. R for right shift, R for Bohr effect. The curve can be shifted to the left also like this. It can go to the left side. That is called left shift of the curve. So left means tissues are left without oxygen. So if hemoglobin is binding all the oxygen, the tissues will be left without oxygen. So hemoglobin oxygen affinity is very high in left shift. Exactly the opposite happens in right shift. What happens in right shift? Hemoglobin is releasing oxygen to the tissue. So oxygen hemoglobin affinity is less in right shift. But in left shift, hemoglobin is holding all the oxygen. Oxygen hemoglobin affinity is more. Left means something bad, no, something negative. We'll place all negative factors. Decrease in H+, decrease in CO2, decrease in temperature, decrease in 2,3 BPG, fetal hemoglobin, stored blood. What happens in stored blood? When you store blood for a long time, glycolysis decreases. 2,3 BPG is an intermediate product of glycolysis. If glycolysis is less, 2,3 BPG also will be less. That's why left shift. And left shift is called Haldane effect. Right shift, R for Bohr effect. Left shift, L for Haldane effect. So I think that was a very quick, rapid revision of the high yield topics for the upcoming INI CET. So please go through all these topics. All these are must learn topics and must revise topics. Almost you will see that even if the questions are not repeated, topics are repeated. So you'll definitely have an overlap of topics. So please make sure that you get hold of all the previous year question answers and also the previous year topics. Yes, with that note, success is not final, failure is not fatal. It is the courage to continue that counts. My best wishes to all of you. We end our session with this. Any queries you can post in the comment box. Thank you so much for joining with me.